Welcome to the Jackson Laboratories Humanized Mice PubCast, where we offer a glimpse into the world of humanized mice through the window of a recent scholarly publication. In this series, we're defining humanized mice as immunodeficient mice that have been engrafted with human CD34-positive hematopoietic stem cells or a human PBMC-derived humanized immune system. JAX is a longtime leading provider of both naive immunodeficient and humanized mice to the life sciences community worldwide. We regularly run preclinical studies for external clients using these models, most commonly in the fields of oncology, immuno-oncology, and transplantation biology and drug development. But we're always excited to see the new and different ways that other researchers use these models. Join us as we explore the cutting-edge research and applications of humanized mice.
Hello, everyone. My name is Dr. Sam Eck, and today we're going to be taking a look at a recent publication from the journal Blood titled CD19-CD28, an affinity-optimized CD28 agonist for combination with glofitamab as off-the-shelf immunotherapy. In this paper, the authors sought to develop and characterize a novel immunotherapeutic antibody that works in concert with an already FDA-approved drug to enhance efficacy for the treatment of large B-cell lymphoma. There are two major types of immune cells we'll be talking about in the context of lymphomas: B cells and T cells. In large B-cell lymphomas, B cells, which are typically a normal part of the immune system, which help create antibodies, become cancerous. T cells, another type of immune cell, play the role of recognizing and eliminating cancer cells. So in the case of B-cell lymphomas, T cells can recognize and eliminate cancerous B cells. One approach to developing therapies for lymphoma is to create bispecific antibodies that can recognize markers on B cells and markers on T cells to synthetically bring these two cell types together and help give T cells the chance to exert their anti-tumor activity against the cancerous B cells. One such therapy that was recently approved by the FDA for treatment of B-cell lymphoma is glofitamab. Glofitamab works by binding to the CD20 marker on B cells as well as the CD3 marker on T cells. Glofitamab essentially achieves what's known as signal 1 of the T cell response. Before going further, let's take a moment to review the process of T cell activation. Complete activation of the T cell is a multi-step signaling process requiring at least two signals. The first is called signal 1. This is an antigen-dependent signal where the antigen-specific T cell receptor binds to the target antigen, establishing initial physical connection with the antigen-presenting cell. So now physically bound to its target, the T cell needs a secondary signal to become active and respond to the threat. One way to achieve this is through signal 2. Signal 2 is generated via cognate interaction or direct cell-cell interaction between co-stimulatory molecules on the antigen-presenting cell and the T cell. Depending on the type of T cell, this cognate interaction can have different outcomes. In this publication, the authors were targeting the co-stimulatory receptor CD28, which is involved in outcomes of T cell priming, proliferation and memory, cytokine production, and target cell lysis. So now with that background in mind, going back to the publication, again, we have glofitamab, which acts via signal 1 to redirect T cells to malignant B cells. The authors of this publication wanted to further enhance the efficacy of this therapeutic, and to do so, they hypothesized that co-administering a signal 2 agonist with glofitamab to achieve the full activation of T cells would lead to enhanced depth and duration of anti-tumor effects. As I mentioned, one co-stimulatory receptor that has been of interest to establish signal 2 is CD28. However, previous iterations of a CD28 agonist have been unsuccessful, with one resulting in life-threatening cytokine release during clinical trials. This was largely due to a lack of sufficient preclinical models to accurately evaluate safety. To circumvent these previous issues, the authors incorporated humanized mice throughout their drug development pipeline. And as we will see by the end of this episode, including these models, which express human-specific targets and accurately recapitulate human physiology and pathophysiology, they were able to successfully develop a novel bispecific antibody that targeted CD19 on tumor B cells and CD28 on T cells. This novel therapeutic works in concert with glofitamab and is currently in ongoing phase 1 clinical trials. So let's dive into their methods. They used a couple of different humanized mouse models in their preclinical drug development pipeline to assess efficacy, safety, and pharmacokinetics. This helped calculate a safe starting dose for humans. A lot of the anti-tumor efficacy and safety testing in this paper was achieved using extremely immunodeficient mice that were humanized with CD34 hematopoietic stem cells, which contributed the source of human T cells. First, in CD34 immune humanized NSG mice, which were co-engrafted with tumors, they were able to show that the combination therapy of their novel CD19-CD28 antibody with glofitamab achieved significantly greater tumor regression than mice treated with glofitamab alone. Second, to evaluate safety, they measured cytokine release in CD34 humanized mice. With these humanized mice, the authors were able to show that their novel low-affinity therapeutic does not elicit cytokine release, thus avoiding dangerous CRS that was seen with a previous version of this antibody. Third, they used a genetically humanized FcRn mouse model to evaluate pharmacokinetics that would allow them to inform human dosing for clinical trials. And this is where we're going to pick up the story today. Before we dive into their FcRn study design and data, let's take a look at humanized FcRn mice and the role that FcRn plays in antibody half-life. In this paper, we're talking about the use of two bispecific antibodies as immunotherapies. Antibodies have become very attractive as therapeutic candidates for two reasons. One, because of their high degree of target specificity due to the variable regions, and two, antibodies exhibit an extended half-life due to the presence of the constant region of the antibody, or Fc, meaning less frequent dosing is needed in clinic. In particular, the extended half-life of IgG antibodies is due to binding between the IgG Fc region to the neonatal Fc receptor, or FcRn. This is present in the endothelial lysosome. This binding rescues Fc-containing molecules from lysosomal degradation and recycles them back into the bloodstream where they can be available to their therapeutic targets. Now, FcRn exhibits very different binding affinities across species, and particularly between mouse and human FcRn. Therefore, to accurately predict PK of human IgG, it's important to evaluate the experimental antibody in the presence of human FcRn. Humanized FcRn mice allow this accurate PK or pharmacokinetic modeling. Now that we have a foundational understanding of FcRn, in this publication, the authors utilized the Jackson Laboratories TG32 humanized FcRn mouse to evaluate human PK. These mice have a null mutation for the mouse FcRn gene and carry a transgene expressing human FcRn. These mice have the most human-like protection of humanized IgGs. Using these humanized FcRn mice and human PK predictive allometric scaling, you can estimate the minimum dose to achieve therapeutic serum concentrations, reducing the need for potentially risky dose escalation treatments during clinical trials. And I want to point out that JAX has several different humanized FcRn models available. I'm including a link in the description to our field guide, which walks through our different FcRn models in more detail and can help guide you in selecting the best model option for your experimental goals. So now that we've set this foundation, let's take a look at how these authors utilized the humanized FcRn platform. They had 8 female TG32 mice, which were administered a single intravenous dose of 10 milligrams per kilogram of their CD19-CD28 antibody. They then collected blood serum at various time points, ranging from shortly after antibody administration to 3 weeks later. They used an ELISA assay to quantify CD19-CD28 antibody levels in the serum samples. And finally, allometric scaling was performed on the data collected to predict human PK and estimate a relevant starting dose for humans. And here we have that allometric scaled data. So in this pink or purple color, we have the PK predictions based on the data collected in the humanized FcRn preclinical study. That's scaled for 3 different human doses. We have concentration on the y-axis and time after dose on the x-axis. And using the preclinical data that the authors collected from the FcRn mice, as well as the preclinical safety and efficacy testing in immune, humanized, and tumor-bearing models, they were able to move forward with the first clinical trials of their antibody. And you can see actual observed human PK data from this ongoing clinical trial overlaid here in the light gray color. So you can see how closely and accurately the authors were able to predict real human PK data based on their findings in humanized mice. And this is really important to not only understand what is a safe and effective dose of your therapeutic, but to also understand how long it stays in the system to inform how exactly it's appropriate to administer your therapeutic, at what frequency, at what concentration, in order to achieve that effective dose. Once again, I encourage you to read through the full publication linked below to see how all of these pieces came together. Thank you all for tuning in for another JAX Humanized PubCast, and we'll see you next time.